Camille gave a nice introduction about absorptive and fine roots, so I'm measuring and analyzing the same subject. And now I will tell you about how they react in relation to changes in soil temperature. And as you can see from the... Yeah, I'm a bit taller than Camille. Thanks, it made my life easier. As you can see, we were interested in how soil warming affects spruce roots. Our study is unique because we had the chance to compare results from an artificial soil warming experiment located in a 130-year-old Norway spruce forest in close to the village of Achenkirch in Austrian Alps, to observations from a natural soil temperature gradient in Iceland. And the man-made experiment warming took place 9 years there, and it was conducted by buried heating cables in the soil. And the warmed soil was kept plus 4 degrees from the ambient. For hot geothermal soil temperature gradient was located in Iceland in a 50-year-old Sitka spruce forest. And there an earthquake took place in May 2008, which shifted geothermal systems to previously unwarmed areas. And as you can see, in the hottest end of the gradient here, tree roots experienced temperatures up to plus 14 degrees from the ambient. This was measured while we were taking samples in September 2013, so the actual long-term averages are lower. In the average temperatures of the hottest warming level on the gradient in 2013 was plus 8, both for the whole year and the vegetation period. And as you can see, well, actually the gradient goes even further or hotter than that, but trees can't endure this heat anymore, and the die-off edge starts here. So we have the unique chance to study how the fine roots react to such warming. This graph illustrates again how soil temperature changes on the for hot gradient, both horizontally from the ambient plots, the A plots, to the hottest plot, the E plot. And because of the geothermal origin of soil warming, then it gets hotter also as the soil gets deeper. So therefore, we measured root morphology also in 3 different soil depths. And for comparison, I've put the Achenkirch warming level here. It's plus 4 degrees, so you can see it's quite moderate compared to the hottest end of the gradient. Camille already told you about fine roots and absorptive roots. We measured similar root traits as she did. So again, shortly, this whole root segment would be considered fine roots if it had a diameter of less than 2 millimeters. It contains both roots of primary structure and secondary structure. And in addition to measuring fine root biomass, we also measured absorptive fine root biomass. So again, we consider absorptive roots as first to third order roots. In addition, we measured absorptive root morphology, diameter, specific root area, specific root length, and root tissue density. And also, we considered branching as part of as a morphological trait, but Camille presented it as an architectural trait. So I believe it's partly a matter of taste. And this is what our morphological samples looked like in the WinRISO image analysis system. And in addition to that, we had the chance to study the colonizing ectomycorrhizal fungi at the Achenkirch site. So again, as Camille said, the ectomycorrhizal fungi largely influence both root uptake directly and not only nutrient uptake, but carbon cost to the host, etc. But also they influence root functioning through changes in morphology. For example, changes in diameter and root length, and this in turn affects these compound traits. A few words about what was known before we started our study. Some other soil warming studies have reported a decrease in fine root biomass or an increase in fine root mortality. The only other soil warming experiment that I know that measured absorptive root morphology similarly as we did reports an increase in specific root length. They actually measured other morphological traits as well, but the effect size and the direction of change depended largely on whether the soil was additionally fertilized and irrigated or not. And the same applies for any fungal community analysis, that their results largely depend on whether the soil is nitrogen limited or, on the other hand, has large availability of labile nitrogen, a number of other factors. And not much was known about how extreme warming affects roots and fungi. Now to the results. We were happy to see that soil warming had a similar effect on the fine roots at both sites. It increased specific root length and area. Yeah, here. So in our case, specific root length and specific root area were in large correlation. Also, the warmer soils had longer absorptive roots, which co-occurred with decreased branching per root length. And also we saw a decrease in root tissue density. As for diameter, then at the Acclimations site, roots were significantly thinner in the warmed soils, but this was partly because of a especially large morphotype present in the ambient, in one of the ambient plots. And also there were some uncolonized roots in the warmed plots, so this might have influenced this. And for hot gradient, we did not distinguish a clear warming effect on root diameter. The last two traits are absorptive fine root biomass per basal area and fine root biomass per basal area. So as you can see, there was a sharp reduction in the hottest plot, +8 degrees for hot gradient. Although in the lower warming levels, the biomass even slightly increased. Now, as I said, for hot gradient, we measured the morphological traits in different soil depths. So while specific root length and specific root area increased gradually in all the 3 different depths in relation to warming, then branching per unit of length and absorptive root length changed significantly only in the 2 upper layers, which experienced more moderate temperatures. So it would be about here. And tissue density started rapidly declining only in the red area here, which experienced the highest temperatures in the 2 deepest layers and in the hottest plot. And we attribute the changes in root length and branching to decreased root tip proliferation. And a decrease in tissue density might indicate increased turnover and decreased lifespan. And this was actually measured at the Acclimations site as well, but not published yet, as I understand. This graph illustrates how the fine root biomass drops to near zero at around 4 degrees from the ambient. And by 6 degrees from the ambient, the trees cannot sustain sufficient fine root biomass anymore. And root tissue density followed a similar decline and correlated most of all these morphological root traits with fine root biomass and absorptive fine root biomass. As for the fungal community, the only group where we distinguished a significant difference between the treatments was the Tomentella genus, which was more abundant in terms of colonization percentage in the warmed plots. But soil warming did affect the community structure. It was more varied, or other words, the distances of the Jotus were larger to the group centroid. And here, red color signifies warming and blue color signifies control. And also, long-distance exploration type was present only in the warm plots represented by Boletus luridus, but since the colonization percentage of it was so low, then we couldn't get a significant difference there. In conclusion, we believe that the similar fine root responses at the two sites reflect general acclimation patterns of spruce in relation to warming, and spruce species seem to respond most actively with branching and root length. And this has actually been shown before as well in a couple of publications of Ivik Ostonen, my supervisor. Also, for hot geothermal gradient, +4 degrees emerged as a threshold in warming endurance, from which onwards the trees couldn't endure the heat anymore. And both the root morphology and the ectomycorrhizal community proved to be sensitive to even moderate warming elevations, even happening at small spatial scales, because at the Achenkir site the warming plots are quite small, and even after 9 years of warming in a mature forest. Thank you.